Thank you, Wes, for that warm introduction. Thank you, Fadi and Wes, for inviting me to this. This is the first time I've been to the Cancer Genome Consortium meeting. I've had an absolute ball meeting a whole bunch of people. I'm inspired by all the technologies that you're using and the impact that you're having for American cancer patients. So I wanted to help open this sort of cancer bioinformatics section by really trying to relay some of the experiences that we've had in Australia doing some precision medicine work for children with cancer. I want to thank my amazing team who without any of this, none of this would be possible. I have no disclosures. So in 2010, I was a pretty young postdoc working out what I wanted to do for my career. I'd just been to a Cold Spring Harbor Biology of Genomes meeting where I met Elaine Martis and she wrote this provocative paper. I could start to see the potential for whole genome sequencing to diagnose a whole range of patients with rare inherited diseases and cancer patients, finding them the right drugs. But that really annoyed me, that the reason that people might not be able to take these technologies or benefit from these technologies was that the analysis was so damn expensive back then. So I tried to do something about it. 15 years later, I've been working on this problem for a long time and probably the rest of my career. But I really wanted to try and overcome this sort of paradox. So I don't need to tell this audience, but I usually like to start with this slide because for decades we've known through a whole range of techniques that you're better at than I am, that cancer is a disease of the genome, and that when we look down the microscope, we see whole ranges of different aneuploidies and horrible marker chromosomes driving lots of cancer. I saw the opportunity, as many have, to apply whole genome sequencing as a single assay that would allow us to zoom out at the cytogenetic level all the way into the single nucleotide-based resolution and reveal remarkable precision around the structural variants and mutations driving each of these different cancer types. I've become more enamored with the power of RNA sequencing, in part through my amazing PhD student, Chelsea Mayo, who I'm going to tell you about a lot of her work today, the power of RNA sequencing to reveal the complementary insights into the molecular basis of children's cancers. And we also think methylation profiling has a huge role to play in refining the cell of origin so that we can answer these key questions that we care about so much for each of our patients. What's the right diagnosis? What's their prognosis and likely outcome? What are the treatment options? And what's the best molecular monitoring strategy? We're also starting to think about whether there's prevention strategies for those families that we identify at risk. So I also really like this slide because whole genome sequencing, many of you will think is a great technique for identifying mutations, yes, with all that sensitivity and resolution, but it also tells you so much more about the cancer genome. Many of you will know about mutation signatures that can occasionally reveal the etiology of that particular child's cancer. I'm really driven by finding biomarkers that we can use to track different patients, whether it's Ig, TCR rearrangements in leukemias or SNP biomarkers for ctDNA profiling, and a whole range of other things. When you put your clinical hat on, you don't have to look at the whole genome. You can look at virtual gene panels and look at the things that really matter fast, but then you can put your research hat on and discover new things about the patients that you're studying. It's really important for childhood cancers because they're still a relatively rare disorder. We're still discovering a whole lot of new disease genes. So yeah, I feel like that's my opening gambit for why I think whole genome sequencing short read is a good test. Right, so my talk today is really going to be in two parts. The first is going to tell you a bit about the Zero Childhood Cancer Program. It's a cancer bioinformatics session, so I've chosen to talk a bit less about the patient impact, but a little bit more about the methodologies that we're using. I'm going to try and be broad. If anything piques your interest, do reach out. I've got QR codes on lots of the slides, which will take you to all the tools and things that I'm going to tell you about. Also, we'll tell you about some things that others have already mentioned about gene expression and methylation profiling to classify tumors, and then how the hell do we put all these pieces together to do this fast in the clinic. So the Zero Childhood Cancer Program is Australia's national precision medicine program for young people with cancer. Australia's got about 29 million people, so it's a bit smaller than the size of Texas. So that means we have about 1,000 young people diagnosed with cancer every single year. We do this rather comprehensive molecular profiling strategy, short read 30x whole genome sequencing for the germline, about 100x for the tumor, deep tumor RNA sequencing with 100 million reads, and these EPIC V2 Illumina microarrays. We've been doing this for more than 10 years, actually, so we've got some insights in bringing these different technologies together. We look at the data differently depending on the type of cancer patient. So for the high risk and rare cancers, we're trying to find, yes, the right molecular diagnosis, but we're also trying to work with our national oncology colleagues to come up with the right personalized treatment plan. We also try and conduct high throughput drug screening with a 152 drug compound library and PDX modeling where possible. And if the child unfortunately relapses or progresses, we do another biopsy and repeat this all again and see how much the tumor changed. But for the non-high-risk cancers, which is the majority, 80% of the population, we know that many have defined treatment paths ahead of them. And so our focus there is on finding the right molecular diagnosis and prognosis and having that information up our sleeve should they unfortunately relapse. Here we very heavily restrict the scope of the analysis to these virtual gene panels. Every single hospital in Australia that sees pediatric patients enrolls patients on the study, and we opened 2 sites in New Zealand in the last 18 months, so we're now international. Zero itself is not a treatment trial, so we try and use these molecular findings to guide patients onto treatment trials, including our own basket trial that we call Optimize. We were very lucky to get, not lucky, we worked pretty hard, but we've received government funding from the health department to expand the program for 3 more years, which we're very happy about. Australian patients are very happy about. But also we now get to expand the program to young adults up to the age of 25 with pediatric type cancers. So we are continuing to scale. I won't talk today about our liquid biopsy, but that's coming online pretty soon too. So the headline results from all this comprehensive molecular profiling is that we find a molecular driver in more than 90% of these patients, unprecedentedly high rates of recommending treatments that are available in the Australian setting, high rates of genetic predisposition, which of course identifies families at risk. 3/4 of these families didn't know that they had a risk variant, so that's really important. And 5% of patients, we can change the diagnosis. Last year, we published on a cohort of 384 patients that we had tracked for over 3 years, I think, that we could double the progression-free survival for those patients that received the personalized guided therapy. So I'm pretty proud of that achievement. But even further, our lead oncologist, Loretta Lau, looked further at the recommendations and the patient responses. And if you had 3 favorable factors, such as the drug having some tier 1 evidence, the driver being a fusion or a structural variant, and the patient received that therapy before relapse, you can see the survival curve tells us we're on the right path. So this is pretty exciting. As I said, this is the cancer bioinformatics session, so I'm going to talk a bit less about the patient stories that I normally do, but really, let's get into it. So we heard yesterday, I think it was Fritz's session, about can you have too much data? Yes, you can. For each of these different modalities, we have to perform all of these different analyses with custom bioinformatics tools and filters, and really we have to know the behavior of each and every one of these tools so that we can get the most out of it. So I'm going to spend probably the next 10 or so minutes going through some of the ways that we actually do some of these analyses. Many of these tools are now available. There'll be barcodes, and you can try and access these tools. So the first thing is the whole genome analysis pipeline. So we've been working with the Hartwig Medical Foundation for almost 10 years now, who are building great software for analyzing adult cancer genomes. We've been adapting that whole time, working with Peter and his team to make sure that these tools work really effectively in childhood cancers. Many of you will know almost every different childhood cancer has a horrible gene that's difficult to study, and we've been putting in stopgaps and fixes into the tools so we can find histone mutations and SS18 fusions and DUX4 pseudogenes and things like that. It's open source, it's available on GitHub. A group from University of Melbourne saw the utility of these tools and spent the effort to wrap all of these tools in a pipeline called a Nextflow pipeline. If you've never heard of Nextflow, it's a really convenient way of wrapping tools together. And if you make it to NF core, that means you've got a really good pipeline that's well tested and robust. So we've been able to influence many national scale precision medicine studies to use and adopt these pipelines. I was talking to Trevor Pugh, Canada is aware of this. England, Germany, Netherlands, Danes are, I believe Genomics England is starting to use these tools. So I'm becoming bold enough to say that this is perhaps becoming best practice pipelines for short read whole genome in cancer. Some little insights into some of the actual tools and why I think they're really effective. Hartwig made the SAGE, SNV, and indel caller. Instead of blacklisting large parts of the genome where you don't attempt to call variation, there's 4 different calling thresholds, whether you're looking for a hotspot or in some of these harder regions. You can append results from multiple tumor analyses, so you can do tumor evolution work. Purple was one of the earliest tools that they built. And if you think about every segmental copy number change, it should be flanked by 2 breakpoints. And so Purple receives those breakpoints as an input and has a precise breakpoint resolution around the copy number segments in each of these cancers. And structural variation has probably been the hardest part to get right as we can. Initially with the GRIDS tool, which has largely been deprecated now in favor of SV. I'm going to show you a lot about this LINKS tool, which I think is amazing, providing insights into these cancers. So this is a pretty sophisticated audience. You know BCR-ABL. This is what it looks like in the textbook with a simple reciprocal translocation. But this seems to be the norm, that as soon as we look at these these structural variants in patients, it's often not that simple. So once you get used to reading these circular diagrams, you can already see there's structural variation linking ABL1 and BCR there. Get used to reading these diagrams and you can see that one of the derived chromosomes links indeed BCR-ABL. This is a reciprocal translocation, so you can read these diagrams in the opposite direction. But I want to draw your attention to just upstream of BCR. Look at all that complex damage happening in the regulatory region of this BCR-ABL fusion. I heard lots of examples of discordance coming from people running FISH assays and not quite seeing and resolving what they're looking at you, Raymond. But this is kind of the norm every time we zoom into these different types of drivers, at least in childhood cancer. So LINKS is extremely effective at also uncovering extrachromosomal DNA. So this is one of my favorite slides. I know it was zoomed out at a long way, but there's like 60 different circular ecDNA molecules, each carrying different oncogenes, driving at different levels of abundance in these different cancers. I've zoomed in here to show you an example of TERT, amplified on an extrachromosomal DNA at about 20 copies. It's probably from a chromothripsis event, because you can see all these other fragments joined in with it. Presumably they are the regulatory elements that are helping drive TERT in this particular neuroblastoma. That's the kind of exquisite level of sensitivity you can get with tools like these structural variant tools. These, of course, matter because extrachromosomal DNAs are poor prognostic factors, and that's something we're really interested in looking into in more detail. So in our 2020 paper, when we looked at all the reportable structural variants, we compared whole genome and RNA sequencing, and only 53% of those SVs were found by both tools. So we thought, let's dig into this in a little more detail. Let's try and close this gap so that both tools find as many of these SVs as possible. So the first thing we did was really simple. Split the structural variants into gene fusions or intragenic structural variants. The intragenic ones are the ones within a gene that might damage 1 or 2 or 3 exons. And so you can clearly see, if you look at red and salmon, that RNA-seq does a pretty good job of detecting gene fusions, as you would expect. There's still a blue bar there, and there's a bunch of gene fusions that RNA-seq missed. And you can clearly see that RNA-seq, with reference-guided methods, the so-called fusion detectors, does a pretty poor job of picking up these intragenic structural variants. So to do something about this, we developed a new RNA-seq mutation detection pipeline called Carbonite. You can see from the colors that we have multiple fusion callers, multiple structural variant detection callers, SNV and indel callers, all sorts of things built into this pipeline. Some of the tools actually receive an input from the whole genome pipeline, so we can guide the RNA analysis to where we think the mutations are from the genome. It's fast, it's open source, it's available now. I'm going to really explain how this works with some examples. So in the top here, you can see that LINKS has resolved presumably another chromothriptic event. You can see the sawtooth copy number pattern here delineated by structural variant breakpoints. And RNA-seq does a great job of resolving all of that complexity and telling us that there's a novel CLIP2 EGFR fusion. The patient did actually receive this drug and had a partial response. Here's another example. We heard about FGFR fusions already at this conference, and here's a moderately complex event. Out of that drove an FGFR1 in-frame activating fusion in a high-grade glioma. So these two tools together are very powerful combinations. It's not just activating oncogenes, of course. Structural variation is a pretty effective way at damaging tumor suppressor genes. This one's kind of horrifying because two really important tumor suppressor genes are simultaneously damaged. Again, you can see this pattern of consistent structural variation and damage. These pediatric cancer genomes are often called low mutation burden. That's not really been our experience when we zoom in with tools like these. The other biomarker that we can use to corroborate that evidence from the RNA is that P53 here has extremely low expression when we compare it to our cohort. Okay, so we can start to use abundance as an indicator of the presence of the fusion or damaging fusion. So to go a little more systematically than this one example, we looked at those fusions that I mentioned were missed by these fusion detection methods. And to make it's not a complex story, but there's really two observations. One is that RNA-seq, as you might expect, does not do a good job of detecting enhancer or promoter hijacking. So on the left here, you can see that there's some promoter and enhancer hijacking events in MYC and TERT, and you can see these red dots that are extreme outlier expression of those genes relative to our wild-type controls. And the other class of fusions that RNA-seq didn't do a great job at detecting were the ones that damage tumor suppressor genes, presumably causing nonsense-mediated decay of the damaged gene. And I'll point in this direction this time. You can see extremely low abundance of some of these oncogenes driven by these SVs. So here we can start to use abundance as the corroborating evidence for these particular types of events. Back to those intragenic structural variants, those events that hit 1 or 2 or 3 exons. These are really common when we use these tools. Over a quarter of our reported SVs are intragenic. We think they're pretty hard to detect using other techniques. We just heard other tools that can. But if you probably can't read it, but that's a smorgasbord of pediatric cancer tumor suppressor genes damaged by these nasty intragenic structural variants. The cartoon here depicts what I'm talking about in a bit more detail, like single exon deletions, single exon duplications, or inversions which become invisible. And it didn't take too long to think about what the signal for these things should be. It's that novel splice junctions appear that miss the exons that are affected. So here you can see a patient where there's a deletion, and all the exons affected by the deletion get skipped. So you get this novel splice junction in the RNA sequencing data. We adopted some reference-free RNA sequencing methods to try and, to identify most of these events. And indeed, using this particular tool, Minty, developed by Alicia Rochlack, you could end up finding the sequence evidence for most of these events. So if we think about that gap that we set out to close, we found all but 2 of the variants with both technologies now. So I think using these tools that are now available, we can kind of detect these mutations really effectively. RNA-seq is fantastic at resolving single nucleotide changes that affect splicing. Here you can see a +5 intronic event that caused exon skipping, or a -1 exon event there, which led to partial exon retention. And so you can really see how RNA sequencing helps resolve the functional consequence of those splice-altering variants. You can also identify variants that are becoming enriched preferentially with allele specificity for the mutated allele, which we take as evidence for the second hit in the tumor, which helps inform the pathogenicity assessment. I think in the interest of time, I have to skip one of my favorite slides. Follow the QR code if you have trouble interpreting splice-altering variants. We've made some amazing tools that I think some of you will find useful for flowcharts and understanding the context of why Splice AI is giving you a high score and you're not sure why. So the QR code's the same on multiple of these slides. Like we've already heard at this conference, you can use whole transcriptome sequencing to classify molecular subtypes of leukemia. We're still exploring different classification tools. I won't make a bold claim that we've nailed this, but we are doing this in real time and having a lot of clinical utility in trying to help diagnose the cancers from these types of analyses. And the last data piece is really the use of methylome profiling, as I said right at the start. Methylation profiles tell you the cell of origin of your cancer, which has had remarkable capability to molecularly diagnose brain sarcoma and neuroblastomas via the German MNP classifier. They produce these reports. It's clinically accredited in lots of labs in Australia. And we've built another tool called Methylyzer, which helps you understand the methylation status of of various loci that we think are important in pediatric cancers listed down the bottom there. So how do we put it all together? It's a lot of analysis, and we're now going to have to ask our disease experts, our clinical our curators, our cancer geneticists, our oncologists, our pathologists to try and somehow assimilate all this information. So in the last 4 or so minutes of the talk, I'll try and show you how we put all the pieces together. So there's really 3 pieces to the puzzle. For about 8 years, we've been running a novel hybrid cloud ecosystem, which helps us use local compute and cloud compute kind of seamlessly. We've built some novel data architecture, which I probably don't have too much time to explain how that works. If you're a data nerd, I'm very happy to have this conversation. But this lets us glue, integrate, and visualize huge disparate data sets together, like treatment data, mouse drug screening with germline variation and all sorts of stuff. And the other thing we spent a lot of effort on is building great apps that help our scientists and curators interact with that data. So one of those apps is called MTB Notes, built by Alan. It's sort of our internal precision medicine knowledge base for childhood cancers. Here I'm just showing you some pages of nothing you don't already know, but P53, the pattern of mutations that we see across our cohort in different cancer types. And importantly, what we said about each of those pathogenic variants for each patient so that our clinicians can have a head start writing the molecular therapeutic reports for them. We have an AI project to try and automate some of this as well. We have disease-specific pages here which characterize the main molecular drivers for each pediatric cancer type. Some of this, I should say, if I was more deeply involved in the CGC community, I might not have built all these apps myself, but these are pretty useful ways of browsing all our information. You can tell from the version numbers that we often review our gene lists in each of these different diseases, which helps power a rapid molecular analysis. The Piece de Resistance is the zero-dash platform that we've built. It's the first one we actually use proper UX designers to help us design and build. And so this is our platform for analytics, reporting, visualization. Like many in the room, we have this challenge of how do you interact with dozens of different data sources from around the internet to help understand the pathogenicity of the individual variants in your cancer. We set out to halve the time it takes to analyze to curate and report each case, and so we built zero-dash to help do that. I don't know if you can see much of the resolution there, but I'm showing you insights into somatic SNVs classified as C5, ranked by our machine learning helium score that floats the variants of interest to the top, the cytogenetics view, so that you can then start to integrate different scales of analysis on this information and how you integrate germline with somatic, with RNA sequencing data. We spend a lot of time semi-automating the molecular reporting so we can get those reports out the door as fast as possible. And in the last quarter, we've also deployed a new module in zero-dash so that we can launch the MTB, Molecular Tumor Board presentations from the same app, where we're linking molecular findings to treatment responses in open clinical trials so that our oncologist fellows all around the country don't have to spend all those hours writing PowerPoint presentations. Very rich clinical information in here that can then be reused. So it's kind of an abrupt stop to the talk. I do have a minute left, so I'm pleased about that. So I did want to sort of remind or leave you with some take-home messages. There's that barcode again. So I think short-read whole genome sequencing, RNA sequencing, and methylation profiling are pretty powerful tools for resolving the molecular basis of childhood cancers. The RNA and methylation data help reveal the cell of origin and molecular subtypes, which my analogy is like the jigsaw puzzle. These help give you the edges of the jigsaw puzzle that help focus your mutation-specific analysis and the pieces in the middle to help solve the jigsaw puzzle that is every single child's cancer. I've shown you how we try and integrate DNA and RNA analysis for mutation detection, which seems to find a driver in most childhood cancers. Perhaps the biggest surprise was how much effort and how large I had to build an engineering team to build some of these data lakes and architectures to do this type of analysis at scale. I can absolutely see a role for long-read sequencing to resolve some of our difficult cases and somewhere where there's just too much genomic complexity to try and resolve. And really been reflecting on all the initiatives that many of you have presented at this conference about how we can better participate in some of the initiatives that you're leading, because I think we've solved some of these problems locally. I feel like we're the only people in Australia doing this type of work, so we're almost under a head down, bum up, get it done, and not really aware of what's been going on around the world. So with that, a huge thanks to the Zero program, especially the leadership of Sheryl. A huge thank you to the patients and children who trust us with their lives in this program. An enormous amount of funders and my team for which none of this could happen. Okay, and thank you.
I'm very pleased to introduce our next speaker, Dr. Sheryl Elkin. Dr. Sheryl Elkin is the Chief Scientific Officer at QIAGEN Digital Insights Oncology Group. Dr. Elkin joined QIAGEN through the acquisition of N01 and led a team establishing a rigorous process for analysis and presentation of scientific and clinical evidence for physicians. Dr. Elkin played a lead role in the development of N01 clinical interpretation methodology and have been a key player in the integration of N01 with the QIAGEN Clinical Insights software. Please join me in welcoming Dr. Elkin.
So thank you very much for that kind introduction, and thank you to the organizers for inviting me to speak to you today. And my disclosure, I am an employee of QIAGEN. So we just heard an outstanding presentation from Dr. Mark Cowley talking about how he's bringing all these incredible technologies to pediatric patients in Australia. And it is just amazing the amount of data and different technologies that he's brought. And one of the things that I've thought about throughout the career that I've had is, how do we bring these capabilities, or at least the information, to be able to help treat patients to more physicians, to help the average oncologist, perhaps the community oncologist, to integrate molecular diagnostics and targeted therapy into their practice. If you would step back with me for 25 years, there were very, very few. We have come an enormous way. It's really incredible. I like this slide because I like the timeline. You can see that back in 2001, we had imatinib, we had Herceptin. So we had, very specific, a few very specific targeted therapies that could be used to really improve the lives of those patients. And at that time, we were starting to see more success in identification of molecular pathways that drove cancer. What we started to see was a lot of repurposing of drugs that were used to treat diseases like diabetes to try to target things like mTOR and other pathways that were there. However, you can see on this timeline the enormous explosion of drugs and targets and therapies that came just in the last 10 to 15 years. And that has also created a fairly significant increase in the number of genes that need to be tested and the amount of information that needs to be understood by the oncologists. So during this period, around 2008, the founders of N of 1, Jennifer Carter and Jun Kinoshita, had the insight that most cancer patients were still being treated systemically, even though research in this field was growing and there were a lot of clinical trials going on in academic centers. And most patients and their physicians, even the ones who were at academic cancer centers, did not have the information they needed to access this therapeutic approach. So they set out to try to democratize, so to speak, the molecular diagnostics in precision medicine. So how did they do this? Their initial approach was to start with the individual patient and the individual physician, meet with the physician, and patient collect history and diagnostic data, and then bring in the scientist, that would be me, research the cancer type, identify pathways that could be targetable, clinical trials, then find the laboratories that could do those tests. There were not very many of them at the time. And they were mainly single gene assays, IHC, FISH, single gene Sanger sequencing, or PCR. We had a nurse that helped us facilitate transfer of tissue from the pathology lab to the testing lab, wresting the slides out of the hands of the pathologists. And then we would collect the test results, review those results, prepare a report that was supported by literature, and review with the patient and physician. So this was extremely time-consuming and does not scale. So what do we do next? Over the next several years, more and more oncogenic markers and tumor suppressors and therapies to target those biomarkers were developed, and the field moved really quickly from single gene testing to mass array. You may remember the sequenome panels of a few hundred mutations, up to starting 50 genes, 100 genes, several hundred genes, exomes, transcriptomes, methylomes, you name it. And as these panels grew, the amount of information coming out of these panels and the amount of literature that the physicians needed to understand grew also quite dramatically. There was just an extraordinary increase in the number of variants and then the number of different aberrations that we would see, indicating an increased need for both bioinformatics and manual labor, the bioinformatic and review and curation of variants, to identify which ones were important and which ones were not. The majority of the tests at that time actually didn't point to an on-label therapy. And so this is a graph showing the percentage of cases. We looked at 100,000 cases over in about 2018 and found that in 16% of those cases, the findings would lead to an on-label therapy if the physician knew that that was what they should do. And that was driven largely by lung cancer at the time. A small percentage had no reportable variants or no variants that were associated with any kind of therapy. But in 60% or so of the cases, there were variants or alterations that pointed to a potential off-label therapy or a clinical trial. And in order for physicians to feel confident to put their patients onto a clinical trial, they needed evidence. They needed to understand what was the literature, what was the evidentiary support that this could possibly be a good thing for their patient instead of the standard of care therapy that's well established. So what do those oncologists need? How do they get this information to access precision medicine for their patients? They need reliable variant classifications. They need matching to available therapies, including on-label, potentially off-label therapies. They need the clinical and scientific data that supports that information. They might need clinical trial matching. In some cases, they need the relevant prognostic and diagnostic information. They need the ability to apply their own level of professional expertise, and they need fast turnaround in order to be clinically relevant. So how do we do it? There are a lot of different ways that different laboratories have approached this problem, and every lab is different, and every lab will have a different approach to this problem, and these approaches are evolving. Obviously, most labs are not doing that one-on-one with the patient and the doctor anymore because it's not scalable. So what is scalable? Manual curation by a team of experts is one approach. It includes variant classification, therapy and trial matching, preparation of a report-ready text. There are solutions that involve automated variant classification based on manually or machine-curated data and logic-driven algorithms. Many laboratories are committed to community collaboration, where there's submission to a common platform that's supervised by trained editors of course, everyone has access to public knowledge bases for variant classifications and descriptions. And we are in the age of artificial intelligence, so we're starting to look at how can we use LLMs and natural language processing and generation to help to classify variants and draft reports with human supervision. So there are advantages and disadvantages to each of these approaches. And which approach is right for each laboratory really depends on what that laboratory is doing. How many different assays do they run? How large are the assays? Do you have a small panel? Do you have a large comprehensive panel, an exome, a genome? What kind of access do you have to professional staff? Do you have variant scientists on your team? Do you have genetic counselors? Do you have bioinformaticians, pathologists? What kind of IT infrastructure do you have? Do you have an in-house knowledge base? Do you have in-house report generation capabilities? So these are all considerations that go into how a lab decides how they're going to approach this question of clinical interpretation. What are you going to bring to that oncologist? So I'd like to discuss a couple of these different approaches. One is the community collaboration and open resources. There are lots of them out there. I've listed only a few. I know there are lots of other ones, so I'm sorry if they're not on the slide. OncoKB Civic, Franklin has a free version, ClinVar. These have the advantages of they're free. They have open access. Everyone can access them. Lots of people can contribute to them. You can leverage the expertise of your community and other experts. Some considerations, they do require review for consistency and accuracy. They might not be comprehensive because the people that are contributing to them are donating their time, and lots of people. How many of you have lots of spare time that you want to spend submitting variants? It's time consuming. And often these tools don't always produce reports. So you can get a lot of information, but then you need to write up the information yourself and generate your own report. There are commercial clinical interpretation solutions that can help with economies of scale. So larger labs that have a much higher throughput may be inclined to go with a commercial solution. Of course, full disclosure, I do work for QIAGEN, but there are many other ones out there. Valsera, Franklin, Roche has Navify, Sophia, lots of them available, and they all have different advantages and disadvantages. As a class, they often have the advantage of a dedicated team of professional curators that are PhDs and/or MDs that spend all of their time, that is what they spend all of their time doing and thinking about, is variant curation. And they do it at scale, allowing reuse, efficient reuse of information. When we first started, we had what my husband lovingly referred to as the Excel spreadsheets of death. These have now, companies and laboratories have now built databases that allow you to escape the death from spreadsheets and efficiently reuse your information. The commercial clinical interpretation companies often have very broad coverage of genes, variants, and cancer types because they see so much, they interpret a lot, and so it's very likely that if it's a common variant that you've seen, that they've seen it before as well. They report in a standardized format and often have report generation capability that's customizable to some extent. Considerations of, again, same consideration. They do require a review for consistency and accuracy. None of these systems are perfect, and they come at a cost. So coming back, what are the clinician's chief concerns? When you're doing this curation and trying to bring this information to the oncologist, what do they want to see? And remembering these oncologists, what we've seen over this meeting, incredible amounts of really complex data. They do not want to see that. They want to see one page, a table ideally, with columns and rows, and what do I do for my patient and how do I read it quickly? So they need accurate variant classification. They need to know what's the actionability, what do I do therapeutically, how does this help with my diagnosis or understanding the prognosis, what are the levels of evidence or what tier is this variant, and all of this information is coming from lots of places. It comes from scientific literature, Cosmic, cBioportal, ClinVar, Nomad, drug labels, guidelines. So how do the people doing this interpretation integrate all of that information? And luckily, this wonderful field has developed guidelines and standards that we can use to help us to integrate that information. So starting in 2015 with the ACMG guidelines for inherited variant classification, we started to have rules and criteria for classifying this information. Marilyn Lee and her team in 2017 published the AMP guidelines that help standardize tiering and levels of evidence for oncogenic variants. And then a few years later, the collaboration between ClinGen, Vic, and CGC, of course, came up with the guidelines for clinical reporting for oncogenicity classification. So all of these things really do help to organize the information and to standardize reporting across the field. But there's still a lot of work to do. So one example of an automated platform that Milena was talking about earlier from QIAGEN, this methodology uses a lot of human curators and some help from, we're starting to develop the use of machine learning to identify papers in the literature, extract information from them, and convert unstructured data into structured data and store it in a knowledge base, and then devise algorithms to execute the rules from the AMP guidelines and the ACMG pathogenicity, oncogenicity guidelines against that data to compute draft classifications. And so using that information that's initially curated by humans but has rules being able to pull in all that information has been can give you something to start with and a place to start and review. A different approach is fully manual curation and classification that's supported by software that facilitates the reuse and efficiency. And in that situation, a scientist will, instead of having the draft classification placed in front of them, which is made based on binary decisions on each piece of data, they'll go out and collect all of the data for a specific variant or a specific gene in a disease, and collect that data, review the papers, and make decisions, and then prepare brief summaries with references so that the physicians can use that as a reference if they need to support those decisions that they're making. So you can use a graphical user interface to manage this information. A VCF can be uploaded subject to pre-configured or flexible filters, and then display the variants color-coded by pathogenicity and actionability with lots of summary information to help the user make those decisions. None of this is a black box, ideally, and I think all of these systems have ways to make a deeper dive into the supporting evidence, and whether that evidence has been pulled in by a human or by a computer program, it is shown, it shows you what rules were triggered, it shows you what pieces of evidence are there, and potentially what was in that evidence that helped draw that, and links you out to that data as well. And a lot of these systems will also help you to generate a report with varying levels of customization, varying levels of detail. But again, for the oncologist, with that really hopefully clear information on the front page in case they rip that one off and never look at the other ones, which you worked so hard to prepare. So what makes this hard? What are some considerations and complexity that surround this clinical somatic interpretation? Data evaluation can be objective, but it can also be subjective. And the guidelines are incredibly helpful, but they still require evaluation of the data to determine if these conditions have been met. Some data is objective. How often you have a KRAS mutation in a pancreatic cancer, you can look it up in Cosmic or CBio or any number of papers. That is objective data. But experimental data requires assessment and analysis. How good is this one particular experiment? What do you do when you have several and they don't agree with each other? And the transparency to that supporting data in the report is critical. We met on Saturday at the unconference and talked about VUSs. Lines between classifications can be blurry. We spent several hours on this topic. I cannot say that we solved it. I wish. And you have to make decisions about which way you err. So if you err on the side of likely oncogenic and take a questionable but very suspicious variant and push it into the oncogenic pile and call it a tier 1 or a tier 2, you do run the risk that someone could be overtreated and treated with a drug that they shouldn't be getting. However, if you err on the other side and the variant goes into a long list of VUSs, then you could, the physician could miss a chance for a successful therapy. And so it's a difficult decision, and it depends on whether you're doing manual work or whether you have a computer that's presenting you a draft list. How do you want to tune your system to error? Because it's not going to be perfect, I guarantee. A few other examples: when you're dealing with drug associations, the levels of evidence can be, as outlined in the AMP guidelines, are very often very clear. So a level A is on the label for that variant and that disease. Level B is a little tougher. What is a well-powered study? What is consensus in the field? It's a little bit more subjective. Level D is clear. It's preclinical data, nothing more. Level C, it's not hard to figure out what it is, but it's hard to convey often how strong that information is. And a lot of physicians would like us to differentiate between high-level and low-level C evidence. When you get into prognostic and diagnostic levels of evidence, it gets much more subjective. Level A is included in guidelines or pathognomonic, but the guidelines often have different levels of evidence that they're using to determine that something is diagnostic, and sometimes it's essential, and sometimes it's preferred. Not everything is created equal. Multiple small studies, do they make it into level B or level C? And that's a difference between tier 1 and tier 2. So they're not small decisions that are being made by these scientists. A few specific examples: you have a variant with clear evidence that it's activating an oncogenic, but it's not on a CDX. Is that a level 1A? Is it a 2C? You have a gene, this is my favorite, least favorite. Gene X is positive on the drug label. So does that mean positive for mutation, positive for expression? Which is you have to go back to the studies and see what was done. It is not a quick answer. And then different methodologies lead to different solutions. So ERB2 amplification by NGS, if the drug label says HER2 expression by IHC, is ERB2 amplification by NGS, should that be tier 1A on your report for that drug? And I will tell you that some people will say yes and some people will say no. There is no consensus on this issue. Similarly, for a new recent approval of telisotuzumab for CMET expression by IHC, what do you do with MET amplification by NGS or FISH? And then again, referring back to the unconference, we had a lengthy discussion about gene fusions and how to manage interpretation of not canonical gene fusions. So say you have a novel partner and slightly unusual breakpoints that maybe remove some portion of the kinase. How do you know how much of the there's no literature describing exactly how much of that kinase domain you need to be present in order to be sensitive to the drug. So how is that done? And again, of course, all of this can be conveyed in words. You can write it in your variant description. You can, as a pathologist or a lab director signing out, you can make a comment. But it's really difficult to convey all this information in tabular format, and that's required, and it's complex, and it's not easy. But you want it to be as accurate and as reliable as possible. Just a moment on automation and AI. It can improve efficiency. Sometimes it doesn't improve efficiency, but it always requires human oversight at this point. Computers have to make, thinking just about automation, computers have to make decisions that are yes or no, oncogenic or VUS, and the data is often more complex than that. So how does an automated system manage conflicting information from multiple sources? It requires manual scientific review to evaluate and weigh each study. So if you ask me, the combination of automation, AI with human review may lead to the most accurate and efficient results as we're moving forward. So to summarize, Improvements in technology and expansion of knowledge has improved clinical options, but has also created an information bottleneck. There are multiple ways to approach this at scale, but the best approach is going to depend on the individual laboratory. The ideal solutions will obviously bring the most accurate information to the physician with the clearest presentation and in the shortest amount of time. And integration of technology with human interpretation and judgment will lead to powerful results. And so I'll leave you with my confused scientist, doctor, pathologist, oncologist. What do I do? Going from, writing one report for one patient that takes 2 weeks to the situation where I think we all now are, where we're producing hundreds or thousands of reports even in a day that are accurate and usable by those oncologists. And with that, I would like to acknowledge my team and thank you for your attention, and happy to take questions with Mark. Thank you.
Great, the session is now open for questions.
That was a fantastic talks, both of you. My question is for you, Mark. So for the Zero program, up until unfortunately recently, the molecular characterization initiative included Australia and the COG sites there. So a couple of questions. One is, how are you correlating? Is there a way to correlate the results that we issue from the MCI? And the other part of the question, one of the things that I think about from the MCI and probably from your program is really to identify what is the best test for that disease, right? Because, as opposed to resourced labs that can do everything, right? The genome, the methylation, and the RNA-seq, some labs can't do that, and we're starting to see from these programs, a large-scale program, is that there are some diseases that need perhaps detection of SNVs and CNVs, but not fusions. That's not the drivers, and so on and so forth. So, are you kind of keeping track of these things in that program, and how are you correlating the MCI results? Fantastic talk, by the way.
Thanks, Yasmin. That's great questions. Thank you. Yeah, the audience might not know, and I don't want to be political, but the COG used to run trials in Australia until very recently, and that meant that some Australian kids were accessing treatments based on COG, and that's no longer the case. So that's really sad for the kids. We've been working closely with Elaine, and we actually tried to work out whether we could use our molecular testing platforms to help enroll these kids on the MCI study, which we decided wasn't a good idea because of consistency.
Yeah.
So I do think we would be able to compare those results if we can work out how to link the patients in the right research setting. I think that would be a very interesting thing to do. I did once look at the testing platforms that you were running on MCI, and there is a bit of a discordance in coverage. And in some diseases, it was low as half of the things that we were finding I didn't think would be covered by those platforms, which was a bit scary. So yeah, I think that's a really important question, because we all hate the idea of missing an important mutation in our patients, right? We really all want to find the drivers. So I think we should do that. It's a primary objective of this current 02 study, which has only been running for 2 years, to work out, of all these big Rolls-Royce platforms that we're running, which is the most appropriate strategy for each different patient. Yeah. So it's too early to answer that question, but that's something we have to. And it is possible and probable that some cancer types won't need all these different analyses. Mm-hmm. I was worried that leukemias may not enroll, because that's a disease where RNA-seq's deemed as good enough. We find lots of things in the genome that we think might not have been with RNA. And so it's going to be an interesting analysis. It's just too early to really answer you there.
It's fantastic. Thank you.
Thank you.
Same, Mark, again, I have a question. I was really fascinated by the extent, as you mentioned, the intragenic structural variants. So do you find biologically that they occur more frequently in certain of the pediatric cancer types? Would you know that or not?
My intuition, I skipped over that part of the slide. It's probably why you're asking this question. Lots of them you all know, IKZF1, BCOR, Icaros, I already said that.
No,
So
I meant, were they in more certain types of tumours?
I think they're across the board. And sometimes DLG2 is really common in osteosarcoma, the exon 8 deletion, but we see it also in some gliomas. So that's the unexpected surprises. And looking at CBL as well, it's appearing in all these the wrong cancer types. So I was working with my colleague Paul Eckert on how we do the functional characterization of these things, but it's often the right oncogene in the wrong cancer type because we're looking. Whether these are VUSs, we're not sure, but they seem to be functional in the molecular testing.
Yeah, oncogenicity of them too, another
Yeah.
headache, right? So,
Yeah.
yeah. Okay, thank you.
Thanks.
Hi, Dana Wolf. I'm just curious, our tools are getting better, and you've put together some nice things to look at this. Do you ever do reanalysis, and when would you recommend that?
Me? You?
Who is that question for?
Yes, often. Every time the reference genome changed, we reanalyze. These tools are updating all the time, so we're finding false positives and misses, and we're constantly going back and forth. So it's a big
Yeah,
part.
I can talk about that too. We do reanalyze these mutations very frequently. Very frequently, at least annually, and they change. As we learn more and more papers are published, we do find that we are making new classifications and upgrading VUSs to likely oncogenic, and also in the other direction when we've had one paper or one body of work that presented a particular gene in a certain way and made us think that a certain region was important, and then new information has come out to suggest that it's not, or that lots of things can happen. But we do go in both directions, and although more often from the US to oncogenic.
The last question.
Yeah, I guess this is for both of you. You know, there's a ton of data on the germline side that physicians misunderstand VUSs. And all kinds of prophylactic surgery and all kinds of things, have been documented to occur. And I just don't know whether there's been as much work done on the somatic tumor side as to what happens with your reports. My experience is the oncologists are a little more likely to ignore them, but I'm just curious whether you are getting feedback. And then I had a different question about zero, but I just want to ask that question of both of you.
Yeah, I mean, it's really hard because you have to make the decision of whether the mutation is going to be on. If it is on the report, then they can find it. Right. And if there's nothing else to do, they might go into that VUS list. And if we provide all the text that says the function of this is unknown, the relevance of therapies is unknown. But beyond that, we know there are anecdotes where people have gone to those drugs, but hopefully they're looking at the ones that are at the top of the report first.
A lot of paper coming out on the perceived utility by the receiving oncologist, positive and negative, so look out for that.
Yeah.
Sharon, I'll have to cut you off. You can grab Mark afterwards. If you could all give a warm round of applause to Dr. Cowley and Dr. Elkin, that'll be the end of this session.